Okay.
Good evening, everyone, and welcome to tonight's webinar. This is the final in our series on genetics and regeneration. And this one is a really important one because we're talking about translating genomic insights into regenerative therapies. So, here we're going to discuss how to actually integrate what we learn from genetics into your clinical decision making and treatment planning. And we'll explore how emerging regenerative therapies can be aligned with individual biology to optimize your patients' outcomes. So this is what we're all here for at the end of the day, and we're going to have lots of great insight from Gustavo. So I'm going to hand over to him now to get started. And if you haven't seen any of our other webinars, then please do send us a message and we can send you the full series. Thanks. Over to you, Gustavo.
Thank you, Chloe, and well, thank you everyone for being present, and thank you everyone for watching the webinar series. So throughout this the past month, we involved mostly on genetics and mechanisms and signaling and so on. So my aim with all that was to introduce you to all the kind of tiny bits of information that compose the final aim, which is treating patients. So in this final one, in this final webinar, this is our fifth webinar. So just as you have an idea of what we have covered in the previous ones, um, in this final webinar, my aim here is for us to go back to understanding the molecular mechanisms in regenerative medicine, but in view of how each one of the mainly used therapies in the aesthetics world and in regeneration in general um actually work. And my aim with that and my aim with the whole series was actually to provide information on how in reality things work so that you have an idea on how to treat the patients, which patients might respond differently due to the presence of certain mechanisms. And then obviously so that you can understand better and always be on top of the very best information possible to any therapy you start applying to your patients. So just so we are on the same page, um, I've brought these, I've been bringing references for you to have a look at. These specifically are kind of final references I wanted to share, but these particularly are not on the same theme we're going to discuss today. Why? Because these are kind of very dense, specific information on omics, which is essentially everything we have seen, so from genetics, so DNA, RNA, and protein, and their quantities, so that you have this in the back of your mind so that you can read these papers calmly. And then we can actually go into what is our themes, which are the real therapies employed naturally in the clinical practice, in the regenerative and longevity practice. So before we go on to the types of therapy that are normally proposed, and I've selected the ones I find more important for you to understand, I wanted to show you a bit of the idea behind what are um the true mechanisms of each one of those therapies. And normally, and this was a question we have discussed in the previous webinars. Normally, the regenerative world starts with controlled injury. So most of the techniques employed mainly for the skin, they rely heavily on controlled injury. And this is very important because we will see in today's webinar what are the mechanisms behind this controlled injury, but also the fact that while we are when we do this controlled injury, we're actually activating endogenous repair systems that rely on DNA duplication, cell proliferation, and obviously to some extent alterations in RNA expression. But as we climb up the biological complexity of the therapies employed, we obviously are adding more mechanisms and more specific mechanisms to the therapy per se. So we start like less biologically complex with the controlled injuries. It's very clear, very direct way of acting and we know for sure that it's going to activate through many mechanisms. We're going to review um, some degree of endogenous repair, but as we add mechanisms such as DNA-based factors, growth factors, or specific growth factors, such as we have in the PRP, tissue preparations, extracellular vesicles, and actual cells being used in therapy. So as we climb up these these biological complexity stairs, we go on adding mechanisms to it. And this is essentially what we're going to do today. We're going to see the therapy and the mechanism, and obviously review a bit how genetics as a whole, not only genetics as predisposition, uh, plays a very important role in defining how these things work, and therefore you will be better able to understand how your patient is responding to a certain therapy. So we are going to start with one that does correlate to both um, injury and also or controlled injury and also growth factors. So it's something with a certain degree of complexity, which are the PRBs we all know and very are now very well employed. So when we talk about PRP, first thing to understand is, well, what is PRP? So for the ones of you who haven't used it yet, PRP is essentially a derivative from the blood of the very patient. So in most of the situations, this is used as autologous blood. So blood is drawn for the patient, centrifuged, and we have a concentrated platelets in the bottom. I mean, as we filter it, we have concentrated platelets in the bottom. And why to use platelets? This is essentially because when platelets are activated, they contain and they also release a certain mixture of growth factors, cytokines, proteins that bind to one another and lead to at least wound regeneration. So this is the biological meaning of using platelets, is the fact that we know they lead to wound regeneration. But obviously, in the very preparations, in the very aim of it inside the regenerative medicine, we're not only looking at wound healing. Obviously, we're doing a scarring, we're doing some sort or some degree of injury to very specific places, but we're looking at the regeneration per se by using this complex mixture of molecules that are released by the platelets. So essentially, when platelet reach plasma, so this is not necessarily only used in the aesthetic context. We have many of those mechanisms which are majorly driven by molecules which are proteins. So if you remember the proteins that have been produced through the gene expression of them, which are essentially the growth factors. So by growth factors, this is something very well known in biology. Growth factors are normally things that have the function of stimulating cells to proliferate. So these are either going to be in multiple types of cells or specific types of cells. If you look at the main components of the PRP, you will see vascular regeneration, so vascular growth factors, and also overall proliferation, so cell proliferation as the PD growth factors, and also the IGF, which is a very general also growth factors, but it's very well correlated to specific cells on the skin too. So these are things that if you look at the scheme below, you will see that the growth factors, they are released from the PRP, so they are present in the PRP and released from the platelets when they are active. And some of them are going to proliferate fibroblasts, some of them are going to proliferate keratinocytes. And obviously, also we have the vascular endothelial growth factors that are mostly going to drive proliferation and migration of cells that are going to do actual vasculogenesis or angiogenesis. So these are all very well directed to the proliferation of cells in general. So we have specific cells being activated now. And the aim of all that is to provide wound healing in the basis, but now we're not doing wound healing, but rather doing the controlled injury healing. So this is it has to be very clear. Firstly, what causes it and what solves it are essentially the communication between the factors being released from the platelets to the cells that are going to be proliferated inside the cells or inside the tissue. So as you see there in this last scheme for this part, one of the main parts of the proliferation or the regeneration of tissues in general is that obviously tissues need blood supply. So whenever wounds heal or whenever tissues heal in general, there's always the need to activate the production of the production or the improvement of the blood flow locally, because otherwise the cells that are being produced are not going to function. So this is a very important factor in regeneration. So regenerative medicine, we often do not consider the fact that blood flow to the local of the regeneration is needed, but there's a very important aspect here in the functioning of the PRP, which is essentially the fact that once PRP enters and the factors are outside a certain tissue, the endothelial cells you see below this tissue, as you can see here, they start to go towards the growth factors we see. So they perceive the growth factors the platelets have released, and they start to proliferate. So as you can see here, they start to proliferate outwards in the tissue until we have obviously cell proliferation happening to an extent, to the extent in which we have actual tissue being formed and actual new uh, um, blood flow being formed or happening in that very tissue. So we have here a multitude of complex thing happening because we have cells altering their status. So the growth factors are actually signaling to these cells here to alter how they are functioning. So essentially, DNA, RNA expression. And later we have cell proliferation, and then we must have here DNA being duplicated and cells proliferating. So both those things are things we have covered before, and therefore obviously these are further mechanisms. These are just previous results from papers, just so you understand that, well, we are generating cells, as you can see here, and as you can see here. So obviously this implies that the proteins these cells produce, such as collagen, we are just marking collagen here, but obviously there are other proteins being produced. They become higher as we increase the amount of the PRP, and this applies to all the types of PRP in the paper. This doesn't come to play, but as we increase the amount of PRP, we are obviously increasing also the amount of growth factors that lead these cells to proliferate, and obviously will lead to more protein to be produced. So as you can see here, as we increase PRP from 1% to 5%, we increase the production of collagen. And this is what you see here. Collagen is marked in red. You can see more collagen surrounding the cells. So it's an important view for you to visualize where collagen is being produced, and regardless of the number of cells, we also are producing more activity in these cells. And this is also a time for us to recall what we have or revisit what we have discussed in the previous talks. This is actually our second talk in which we discussed RNA expression. So whenever we talk about, well, collagen is being produced, a growth factor is being produced, this always has to be in your mind that the sequence of the DNA, let's say that if here were the collagen gene, if here were simply the collagen gene, we, if we are if we are increasing the amount of collagen being produced, it has to be in your mind that for some reason, for some specific stimulus, the RNA polymerase that binds close to the gene of the collagen molecule, it has to go to the DNA and produce the RNA. And this normally happens through the fact that the transcription factors, which is the process happening here, it becomes active, and therefore it activates the RNA polymerase too, and it does produce collagen. And it obviously has to do with the fact that um, inside the nucleus, we have transcription happening. So we have the DNA inside the nucleus, compartmentalized inside the nucleus. RNA is being produced here as well, and then it's exported from the nucleus to the cytoplasm of the cell, where we have translation. So collagen is essentially produced there. And so collagen is essentially produced there outside the nucleus, and then it's exported from the cell as well. And this was what I wanted to talk to you about. One of the methods that include many or a multitude of mechanisms from molecular biology, which we have discussed, and then we can move on to DNA essentially. So when you talk about polynucleotide, pDRN, and so on, we're always talking about DNA. So the first and foremost, which is not going to be the main theme of our discussion today, is the fact that when we have DNA, so for the very molecular chemical structure of DNA, it's always important to remind you that around the phosphate binding or the phosphate bonds are in the backbone of the DNA, which is represented here in blue. Since we have these oxygen here, it makes it more easy, or it makes it easier for water molecules to go and stay surrounding it. So this is the principle of hydration anywhere, so in any component. And it's also the principle of hydration when we inject nucleotides of any sort to the skin in view of volumizing or simply hydrating it, or it's used topically for hydration. So the hydration is something inherent to the molecular structure, so the chemical structure of the polynucleotide. But this is not going to be our focus today. Our focus is essentially DNA as signaling. So the molecular structure is the hydration. Signaling is what we're going to discover today. Obviously, DNA has been seen here in our talks as information. So obviously, when I tell you, well, collagen needs to be produced through the expression of RNA, this is dealing with DNA as information. What we are talking about today is the use of DNA. It can be used by its structure, simply as hydration, but it also can be used due to the possibility of signaling outside the cells and then inside the cells. What happens? So when nucleotides are free in the tissue, this is a normal process that happens in the immune context of our bodies, but obviously here we are employing it therapeutically. So whenever we have DNA free roaming around a certain tissue, we have two things happening here. One of them is the salvage pathway. So the salvage pathway here is a bit of a simplistic view of the usage of polynucleotides to the tissue, but it does have some truth to it, which is the fact that simply when we introduce nucleotides to the tissue, we are going to add nucleotides that can rebuild DNA upon a DNA repair system. Also not our topic here. Our main topic here is going to go to the adenosine receptors. So whenever we also have DNA free, it is also important to remind you that part of this DNA, one of the bases of the DNA, is the adenine or the adenosine. So the adenosine we have signaling in the bodies, both in our brains and also peripherically, it's the same molecule we have in the DNA. So when DNA is chopped, what happens is that it's going to bind to the A2 receptor or the adenosine A2 receptor. So it initiates a G protein based reaction that leads to AMP conversion and therefore activation of the kinase A protein and also cell proliferation. Obviously, this is going to lead to many other fates. So essentially, we will have more cells proliferating. So we have angiogenesis, we have increase in production of cells and collagen. So this is one of the aims, because we are essentially linking to one specific receptor that is bound to this. And it has also been shown to control inflammation, but this is a slightly different story. But why all these things happen? This happens because our body normally, whenever we have a normal injury, and also whenever we do responses to um to pathogens, so mind you, so why does something happen? It has to do with what would happen endogenously in our body. So what I'm telling you is that in a normal injury, if you cut a cell, if you cut your arm, cells get destroyed and you have DNA being released. The same thing happens for different reasons also when a pathogen enters and we have both cell destruction and also cell apoptosis happening due to the normal function of certain of the leukocytes. But let's stay with a normal injury. So let's say simply our body was injured. Cells were broken, split, and DNA gets delivered to the tissue. What happens is that these extracellular nucleotides, so they were initially compartmentalized inside the cells, they are released, and when they are released, they bind essentially to the adenosine receptor, reminding you that adenosine is this guy here, which is essentially part of the DNA, normal part of the DNA. When it binds to the adenosine receptor, what happens is that it starts this AMP dependent pathway that leads to cell proliferation. So part of how cells proliferate, migrate, and produce proteins depends on the fact that if a cell is injured, or is linked to the fact that if a cell is injured, DNA gets released, binds to a certain receptor, stimulates this same pathway. And obviously, there are many other pathways that lead to the same thing. So we do have as the WNT pathway, the hyposignaling, the hedgehog signaling, all of them are other mechanisms that lead to cell proliferation. But now we have one mechanism that depends on nucleotides and specifically adenosine. But obviously, if we're injecting polynucleotides to a patient's body, one of the things we expect to happen is this process to happen dependent on the receptor of adenosine. So this is a very important aspect of how this works. And that takes us to controlled injury. So we have been speaking so far about injuries that haven't happened fiercely. So they were very, very, very controlled injuries. So we just do a single injection or multiple injections, but very mild. But now we're starting to talk about a physical damage being caused to the tissue. So we're no longer talking about very you know pinpointing injuries, but rather a more extensive injury. Although it's controlled, this is going to start to be now a more extensive injury. So we first started with PRP, PDRN. So all those are things that are injected locally in very localized situations in the body. And we already do have the introduction of a molecule or several molecules that lead to cell proliferation. Now we are dealing with lasers and any other sort of controlled injury. I'm going to focus on lasers, but bear in mind that this is going to be true, majorly true for most types of controlled injuries. What happens for laser for most of the situations that we are either going to ablate a layer of the skin, or we are going to very specifically in the fractional laser. So it's probably the most employed one for regeneration itself. We are going to do focused damage to focused parts of the skin. Why is this important? Why is it worth commenting? Firstly, because, well, regardless of we ablating tissue on the top, or if we are kind of either injecting a needle or just lasers penetrating, um, cells will need to be formed around that anyway. So we will need DNA replication to happen, and we will need controlled cell proliferation to happen in that tissue. So these are things we have from our past webinars. So the DNA proliferation or the DNA replication and cell proliferation is fine. It must happen. So it will happen anyways. But the reason why these things happen and how these things happen is also interesting to see. I've brought you, and I would expect you to check this paper later because it's a very interesting one. But I'm going to summarize the most important thing here. Um, we've covered DNA and we've spoken about RNA when we're talking about PRP here. Um, but now it's important for us to kind of visualize very specifically how this happened. What is the dynamics that lead this proliferation to happen? What is the dynamics that lead the tissue to actually form anyways? So let's say we've done this injury to the skin, and this is essentially what happened here. And then across days, tiny bits of the skin was acquired, and we were looking for alterations in which RNA molecules are present and which molecules and which RNA molecules are more present, so as to say. What does it mean? It means that when we move on from the day 1 until the day 28, this is the same image, it's just focused in different molecules, okay? Uh, but when we look at specific molecules or specific RNAs being produced, what happens is that in early days, so from 1 to 3, we see a pattern that is mostly unchanged. So we don't see much of a pattern here. I know this is going to be kind of an abstract thing for you. So I'm a geneticist, I look at these things more often, but you won't see much of a pattern happening here. We have specific genes being expressed here, which are the lines. But you'll see that in the first days, you will see something happening, but not much of a pattern happening. What is very interesting is that obviously we will have alterations in the metalloproteinase protein. So this is a very interesting finding here. But other than that, it's just stressed. So the tissue has just been stressed. Obviously, the metalloproteinases are being expressed more in these days. This is very interesting. However, the most interesting thing happens when we look at from day 14 to day 28, in which we start to see when we focus in genes such as collagen, the metalloproteinases to all the types of collagen, are the spark protein, and so on and so forth, which are all proteins related to the building of the tissue per se, we see that they become red. So that means essentially that this is when these proteins start to be more expressed. So if you remember what we talked about gene expression, this means that the damage that happened in the beginning, regardless of the expression of genes, let's just focus on the damage that happened in the beginning, while it recovers, so as it recovers, we have some stimulus that lead to not only cells proliferating, but it leads also to transcription factors linked to collagen, to metalloproteinases are dealing with the tissue, and so on and so forth. All of that are indicating that fibroblasts have been activated, that proteins have been produced, and overall tissue is being produced, or tissue is being recovered as a matter of fact. So This brings me back a bit now to gene expression. So this is how gene expression is, viewed from a scientific perspective, kind of a very direct way of looking at gene expression. But this also brings me to reminding you that, well, in the past we have seen the mechanisms related to how DNA is transcribed to RNA. But in certain situations, the type of gene expression that happens, or the types of alterations in gene expressions happens in different manners. So a normal thing would be that a tissue that is always dividing or always doing a certain type of metabolism, we see gene expression of two different genes just going up and down. So we need more of that protein, that gene is more expressed, we need less of this, gets less expressed. We already know the mechanisms that control it. However, when we have a um, when we have something that is an impulse, so let's say a laser or something happening to the tissue, a shocking thing happening to the tissue, we have a mechanism being activated that leads to very large variations in gene expression. So what I'm showing you here is essentially that we do have, or our tissues have already, um, the mechanisms that are behind the fact that, well, we have suffered in a certain injury. When this injury happens, the cells respond this way in order to repopulate and also to rebuild the tissue. So this is what you are seeing here. We have suffered an injury, we have suffered a certain injury. When this injury happens, the cells already know what is going to happen, so they already know how to respond, and they respond primarily um, across a certain time by changing how much RNA is expressed by the cells, and then obviously how this RNA is going to alter the functioning of the tissue per se. This is just a kind of a better view of what is happening. So if lasers enter, they're going to leave this coagulation vaporization zone. So they essentially leave a tiny, teeny hole there. And what happens later is that we have all that process of RNA expression happening, in which we have initially the metalloproteinases type 1 are increasing, later the type 2, and also obviously responses to heat and the responses of the transforming growth factors beta that lead both to the organization of the immune system around the tissue, but also to the fibroblast proliferation that eventually will lead to protein production and the reorganization of the tissue as it is desired by this controlled injury. This is just so you can visualize this also microscopically. This was real in vivo microscopy done to laser damage. So essentially, very early in the laser injury happening, what happens is that we have some degree of proliferation of the immune cells, and they start to drive all those changes that happen in RNA expression, collagen production, and so on and so forth. So it's essentially a response to damage, but a very controlled response to damage. That takes us to fat-derived approaches. So the last three approaches we're going to discuss today are mostly cell-related. So they're going to be up in the scale of complexity, essentially because they either deal with cells or they deal with compounds coming from cells directly. Well, so fat-derived approaches, back in the day, so when I was a student, and I did my kind of research assistant thing, and then during PhD, I kind of changed focus. They're the real photos of um, how fat tissue becomes cells, or how fat tissue can be managed inside the laboratory for us to extract cells from them. There's a very big trend, or there was a very big trend on using the stem cells we have in the adipose tissue because, as you know, the adipose tissue is very good in proliferating, in vascularization, and so on and so forth. So there's a big part of research not necessarily focused on regeneration for aesthetics purposes only, but regeneration in general and longevity and the understanding of stem cells. And it depends on um, essentially getting the fat tissue. This was not very usual to get like tissue as a whole, but I thought it was better to show it like that. It also may come from lipoaspirates. What happens is that once we have the fat tissue in our hands, what we do is that we cut it and we put it in contact with a protein called collagenase. So we are breaking the collagen. And as we break the collagen, we put this product of the digestion of the adipose tissue in a tube. And when we centrifuge that tube, you won't be able to see very clearly here in this one, but in the very bottom of the tube, you have this. So this is something for you to have in mind whenever you are dealing with adipose tissue applications, is that if we destroy the tissue or the tissue integrity of the adipose tissue, so we no longer have tissue or cells attached, the fat itself goes up as we would have with oil in the water, and the heavy cells or the most important cells we call the stromal uh, fraction, they go to the bottom. In the bottom, we will have cells which are essentially endothelial cells, both mature and premature, and we also will have stem cells. So they are going to be either precursor cells or stem cells per se, adult stem cells in the adipose tissue. So some of the fat tissue applications in regenerative medicine for tissue regeneration, they do rely heavily on the volume the adipose tissue occupies, but also bear in mind that you have endothelial cells are present there, stem cells present, and they are leading also part of the regeneration that lasts longer by that tissue. So obviously we always need to have in mind that we are dealing with adult stem cells present there, and they are able to repopulate many tissues. In the case of the adipose tissue, they are actually very potent. They are mesenchymal stem cells. But what is important here is for you to have in mind and to differentiate what is done with the adipose tissue. So the simplest method is essentially looking at the adipose tissue in a conventional fat grafting, in which it's simply going to replenish volume loss. So bear in mind that we are replenishing volume loss, but there are cells there that also might replenish or redo the tissue per se. Then we have the more advanced applications that are done in certain situations in which we have either the stromal fraction which is below, and it will have stem cells and also other types of cells that will help um, actually help rebuild that tissue. And that's also important that when it's injected, we have a higher retention of the cells in that tissue, so that that volume that is replenished or the tissue that is being rebuilt is actually going to be it's actually going to last longer in that position. And obviously, if we are dealing with cells that have been culture expanded, so cells that actually have been isolated as adult stem cells, we have an even higher regeneration happening in that situation. Now we're moving on to another cell-related topic, which are the extracellular vesicles or exosomes. These are two concepts that are intertwined per se. I'm not going to go into the detail of the chemistry of them, but bear in mind that extracellular vesicles and exosomes, they're mostly are different in size or just tiny alterations in their composition. What is important here? Um, Exosomes or vesicles per se, they always will be things that are produced inside the cells with a membrane. So you are seeing here some things, they are entering this membrane or an organelle inside the cell that is membranous. It enters a certain amount or certain extension of that membrane, and then it gets expelled from the cell, gets kicked out of the cell. So this is a vesicle or this is an exosome. The process is going to be very similar. The important thing is that something has been produced and has been included in that vesicle. And this is going to either directly go to another cell and sign out to that cell directly simply by binding a receptor outside the cells, or in some situations, it might enter the cell and deliver the content, either RNA, proteins, antigen. This is going to be how these things make communication between cells. And essentially, what we have in the basic biology is that both in delivering information, inside the body, but also in the progress of certain diseases such as cancer, autoimmunity, and so on and so forth, these vesicles, they always will carry something from one cell to the other. So this is the only thing you need to know to understand our exosomes until you go into one specific exosome that has that one particular RNA or one particular protein that leads to one particular um, mechanism happening. For you to know, or for you to have in your mind, whenever someone sells exosomes to you, is also to understand how it is produced. For those of you who haven't had the joy of cultivating cells, which is probably the thing I like the most in science. This is a cell culture flask. And in most of the situations, for most of what is done in science, either for stem cell therapy or for testing drugs or for testing a certain mechanism, what happens is that the cells, they either are suspension, which is mostly for immune cells, or they are adherent cells. What do I mean by that? So this is the cell culture flask, and what the liquid you see in red is the culture medium. So this is a very important thing for you to have in mind. The cells, most of them are going to be cultured, filling the bottom of this plate. This means that these cells are here, so they are in the bottom of your plate, like that. So this is how you visualize these cells. And whatever these cells are producing, let me just change colors here. And whatever these cells are producing, they are exiting the cell and they are going to the culture medium. So there are two very important things here for you to bear in mind. One of them is, well, how does this happen? Is that when we are aiming at producing a certain type of exosomes, we simply will grow the adequate type of cells, and we will expect these cells, we are going to stimulate these cells adequately, and we expect these cells to produce the exosomes and leave them in the culture medium. So what is done to acquire exosomes is not to get cells, but rather to get the medium where these cells are cultivated. Why is this important? Firstly, is that, well, obviously, when you are buying exosomes, it's very important for you to know that exosomes, the real exosomes, they always will come from cell cultures. So you need to know from which cells these exosomes come. So the first thing you need to know. And the second thing, and this is a very important aspect of the clinical practice and also regulatory purposes of the use of exosomes, is to know which type of culture medium is utilized in those cells. So for some situations, some components in those cell culture mediums, they are not approved in certain countries. So it is always important that before buying exosomes or before applying them to patients, always bear in mind that you need to know which cells are in the bottom and which is the composition of the medium these cells are being cultured. So these are two very important aspects of exosome utilization. So let us see now exosomes functioning so that we can finalize that. So this is one also very interesting clinical paper in which exosomes were produced, and this is the size of an exosome. So they are smaller than a cell, so most of it is below 30 nanometers or 30 micros. So essentially, this is an exosome. So this is smaller than a cell. Very important to understand. When they are applied to skin, so they have been applied and they have been very successfully applied in even topically improving um, wound healing. And by improving wound healing, it really improves how cells proliferate in the skin very much. And so you can see here both improvement in in the wounds per se, and also in the texture of the skin. So atrophic scars, they become better organized. Skin pores become less visible in general, skin roughness, but probably the best application and the kind of clearest application is the use for skin healing. So skin healing, and if you expect skin to heal, you also obviously expect skin to proliferate and to reorganize the tissue. And this is a very important aspect of our exosomes being employed. Then we go to our final discussion here, which are the cell-based therapies. So we are moving from the cells product to cells per se. I won't go too much into cell therapy because there aren't many approved cell therapies that really contain cells. So this is something for you to also bear in mind. So before actually using them, you can ask me, we can discuss this very closely. However, obviously, although there aren't many cell-based therapies employed yet for regenerative aesthetic purposes. Obviously, regeneration in other senses have been very well addressed. This is a paper I published many years ago in which we had, or we did, heart infarcts in mice, poor things. So picture the situation. Um, we had mice that had lost cardiac tissue. So the cardiac tissue of these mice had been lost. And we injected through the tail of these animals stem cells. And these stem cells, they have been marked with these things called superparamagnetic iron oxide nanoparticles, which are essentially the green things you see on our screen. What happens here? So in the first image, we had a 12-hour heart tissue being collected. So some of the mice were euthanized, and we collected tissue from the heart of these mice. Why is this important? We injected it in the tail of the mice, and we could track the stem cells going to the very cardiac tissue. So for those of you who remember histology well, this is very clearly cardiac tissue. So the muscle cells in this format are cardiac tissue, and we see stem cells being deposited to the tissue. So what happens here is that the cardiac muscle injured, its signals so cells go to it. So its signals so stem cells know where to go. So this is one of the most important things to understand in stem cell and cell-based therapies. After some weeks, I think this was 2 weeks, so this is from 12-hour to 2 week, we don't see many cells left. So this means that we have some cells left, but the muscle was recovered. So what happens? What happens is that there's a dichotomy in any cell-based therapy. And this is a largely off example for you to understand, but the important thing for you to remind here is that there's a dichotomy and in logic in any cell-based therapy as to whether cells go to the tissue and repopulate the tissue, or they simply go to the tissue and reorganize the tissue. I tend to go more towards the second situation in which we deliver cells to tissues And the same thing goes for the adipose tissue we have discussed earlier. We deliver cells to the tissue. The stem cells, they have certain biological factors that lead these cells to both proliferate a bit, but also to reorganize the tissue per se. So most of what we expect to happen during cell-based therapies is not cell proliferation per se, but rather the organization of the tissue, the signaling in the tissue happening by our stem cells or by the cells we are delivering. And this is the logic of the utilization of stem cell therapy in regenerative medicine. Obviously, there's a very important application for skin, as you can see. So wound healing relies very heavily on resident stem cells happening. And obviously, if we deliver stem cells as it has been attempted, the wounds or the injuries or the controlled injuries that happen to that tissue, they will stimulate the presence of the growth factors we have discussed, the metalloproteinases, the cytokines, and they will stimulate cells to both proliferate and reorganize the tissue. So this is kind of the final thing I had in my mind to tell you with respect to stem cell therapy, so that we can kind of close this with with a final remark. And this is going to be my final message to you. The next slide is just going to be a summary for you to have in your mind. Regardless of the stimulus, so we might have many types of stimuli as we have discussed today. So direct signaling from one specific factor or a sum of specific factors from PRP, or if the stimulus comes from injury or from specific exosomes and so on. There's always going to be a connection between this stimulus and DNA duplication, RNA expression, or a certain receptor being stimulated and signaling between cells. And obviously, all these things they connect too. So when RNA expression changes in routines related to DNA replication, then DNA replication happens. When a certain receptor is activated, we might have more DNA replication, as well as we will have alterations in RNA expression. So um, if I could leave a last message for you from all of those webinars, and also whenever you want to discuss any of those things, I'll be more than ready to and happy to discuss them with you. Whenever any of those things happen or any therapy happens, there always will be a logic behind it. So my aim with all those webinars was actually to guide you into understanding the kind of minor details which are major, which leads to major results in how therapies work. So whenever you start any new therapy, whenever a new therapy is introduced to the market, always look for the patterns of RNA expression, receptor signaling, and DNA replication as a whole, so that you can understand and deliver better treatments to your patients. This last slide is just so you can kind of screenshot and check where these mechanisms go. So the ones we have discussed today, so from controlled injuries until cell-based therapies, where they connect and also where they will act in your patients. So thank you very much. And I hope you have enjoyed. And obviously, I put myself available for any discussions you might need.